Okay, welcome to Let's Get to the Marks. I'm going through GCSE Biology higher tier, paper two. The time allowed is 1 hour and 45 minutes. What have we got here? We've got 9 questions. This is for the AQA syllabus, paper two, 2022, hour and 45 minutes, 9 questions. What I suggest you do is press pause on the video at the start of each question and then unpause and watch the explanation. All right, question one. There are two types of reproduction, sexual reproduction, asexual reproduction. Question 1.1. Complete table 1 to compare sexual reproduction with asexual reproduction. Write a tick in the box if the statement is true. The first row has been completed for you. You can see here it's worth 2 marks. Cell division occurs in sexual reproduction and asexual reproduction. Now you've got to decide on these next 3. Why don't you press pause on the video, have a go on a piece of paper with a pen, and then unpause and watch the explanation. All right, I'm going to go through the answers here, folks. So what I'm going to do here, fertilization occurs in sexual reproduction. Look, in sexual reproduction, you have a sperm. I mean, not necessarily a sperm, but you have a male and female gamete coming together. And so, yes, fertilization occurs. Whereas in asexual reproduction, you just have a cell, no gametes, you have a cell cloning itself. So fertilization does not occur. Genes are passed on from the parent to the offspring. So when the sperm and the egg come together in sexual reproduction, they do go on to make one diploid cell over here that does have genes from both the parents. And in asexual reproduction, you've got all the genes from one parent. So yes, genes are passed on in both to the children. Offspring are genetically identical to each other. So no, because each of the offspring, I mean, it can happen. It can happen if you have twins. You can have genetically identical offspring, but generally no, because it will be a different sperm and egg that will come together to make a different offspring, i.e. brothers and sisters. So I'm going to put a cross there. No, they're not always genetically identical. In fact, rarely. Whereas here, yes, they're going to be genetically identical to the parent, but also genetically identical to each other, as there's only one parent. And so you're looking at tick, tick, cross, cross, tick, tick, and that will get you, oh no, it's not 3, excusez-moi, 2 marks. Boom. Right, we're going to move on. Okay, so question 1.2. Gametes are formed in sexual reproduction. Name the male gamete formed in flowering plants. So why don't you pause the video, have a go in your head or write it down. We just talked about the male gamete in animals is sperm, and that joins with the egg, which is, or the ovum, which is the female gamete in animals. What about plants? The female gamete is called the ovule or the egg. Now, the male gamete is actually something called pollen. Don't know what that's supposed to be. There's some spikes on the pollen. And it's pollen. Yeah, simple as that. Right, I'm going to sneeze. Okay, interesting. Pollen. There we go. Right, let's move on. Okay, we've got a little graph question here. Cell division by meiosis forms gametes. Figure 1 shows the mean mass of DNA per cell before, during, and after meiosis. Mean mass of DNA per cell in arbitrary units. Okay, so AUs, and you've got 0, 1, 2, 3, 4. So it means it's not being measured in grams or a normal mass unit. We just know that if it's 4, when it's 8, it is double the mass of 4. When it's 2, it's half the mass of 4. So that's all these units tell you. They're not in a specific unit. Okay, time in hours along the bottom. Use the rewind. Use information from figure 1 to answer questions 1.3 to 1.6. So they're going to be about this graph. So we're going to do a little bit of coming back up and down here. So let's zing. Now, don't forget, this is meiosis. So just to draw your attention to this, this is meiosis. And I like to think of meiosis, the E for eggs and the S for sperm. This happens, meiosis happens when you're making sperm and eggs. And just as a little clue, sperm and eggs have half the normal number of DNA or chromosomes. They're called haploid cells. Anywho, I've probably given away some of the answers here. Let's go and have a go at the questiones. 1.3, when is the DNA in the chromosomes being copied? Tick one box. Is it between 0 and 3 hours, 3 and 4 hours, 4 and 5 hours, or 5 and 6 hours? What we're going to do is we're going to go up to there and see when is the DNA in the chromosomes being copied, and it's worth 1 mark. So where is it that it looks like? So this is the start before meiosis, before cell division. And then we can see the mass of DNA is 4. Look what happens here. It zings up to 8. So what do you reckon? Pause the video, have a go. I would say it is doubling between 3 and 4 hours. And that is when we're copying the DNA. And of course, that would mean that you start with 1 bit of DNA and then you end up with double the amount of DNA. So you would expect the mass to double. And that's what it does between hours 3 and 4. So that's the one we're going to tick. It doubles between 3 and 4. So, and that is one of the answers. Look, there's our answer right there. Easy peasy, pudding and pie. Right. Let's go down to 1.4. Cells divide twice during meiosis. Which two times in figure 1 show one cell dividing into two cells? It's worth 2 marks, and you've got to tick 2 boxes. So which two times show when one cell divides into two cells? Let's go up and have a look, and I'll try and talk you through this graph. So think about it here. Right now we have one cell. There's my cell with its nucleus. Now it doubles. It's still one cell, still on one cell. Okay. It's just got double the size of the DNA now because it doubled its DNA. So the nucleus is, I've shown that as getting bigger. Now it splits. It goes back to each cell having the correct amount of DNA. So it has split. So this is my first division, and that happened at hour 5. Now, when did it split again? Well, it split again, and now it's going to split into 4 cells. And that's what you get with meiosis. You get 4 haploid cells, each with half the number of chromosomes, which is going to be 2 in this case. And that happened at 6 hours. So I would say the first division happened at 6. The first division happened at 5, the second division at 6. Let's just check what the answer said, because between 5 and 6, obviously they were dividing. And then, yeah, let's go and check it out. What are they actually asking us? Would be nice. So which two times show one cell dividing into two cells? So we're going to say it happened at 5 hours, and 6 hours is when we had cell division going on. So we're going to go there, and that is going to get us. So that's when cells were dividing. They're the 2 divisions. What is the mean mass of DNA in arbitrary units in a sperm cell? So the mean mass of DNA in a sperm cell. Look, in that diagram above, it started as 4, the mass. Then it doubled to 8. Then we had the first division where it divided right into 2 cells of 4 each. And then we had the second division where they divided finally into their, let's say this was making sperm. It could have been making eggs if it was in the woman's ovaries. If it's in the boy's testes, it's making sperm, although we might be talking about pollen in a plant, in which case it's not sperm and it's being made in somewhere called the anther. You don't need to know that. So there's my sperm. Oh, wait, they wouldn't be 4, would they? How can 4 divide into 4 and 4? It can't. So these need to be 2s. Oh, you bell piece, Tom. Right, oh my days. Come on, son, sort it out. Right, 2, 2, 2. We got another one there, 2. That makes sense, right, guys? Hopefully that makes sense for you. There are 2 divisions in mitosis. You've got division 1, division 2. First of all, you've got the doubling. We call this the interphase or the growth phase, and that's where the DNA doubles, and then it splits, and then it splits again. So the mean mass of our sperm cell is 2 at the end. Lovely bubbly. Okay, let's move on. We're on 1.6. What is the mean mass of, oh, we did that. Oh my gosh, I think I'm going bonkers. What is 1.6? What is the mean mass of DNA in arbitrary units in each cell in an embryo? So think about it. Here is our lovely sperm cell with a mean mass of 2. It's going to join to an egg cell that was made in the exact same way by meiosis. So it's going to have a mass of 2. They come together and they make an embryo cell of 4. Now, the embryo cell then doesn't do meiosis. It does mitosis, M-I-T-O. SIS, and they just copy each other by cloning. Okay, so all of the cells in the embryo will have a mass of 4. And that's the final one on this question. And by the way, human sperm cells have 23 chromosomes and egg cells have 23 chromosomes. They were talking about mass here and in AUs, arbitrary units, which means it's not really grams, kilograms, picograms, or whatever. Okay. Earthworms live in soil, feed on dead and decaying plant matter, have moist and soft skin, exchange gases through their skin, give 2 abiotic factors and 2 biotic factors that could affect the size of an earthworm population. It's worth 4 marks. Grab your pen, grab your paper, bit of scrap old paper, grab a pen, press pause on the video, have a go. Well, I'm going to go through the answers. So abiotic factors, what does that mean? Well, abiotic, guys, means not alive, non-living things in the environment that can affect a population. Things like sunlight. It's not a living thing, sunlight, but it can affect plants. Things like water, it's not a living thing, but it can affect plants. Things like carbon dioxide level, which plants need for photosynthesis, can affect plants. Biotic is living. So we're talking about things that are alive, like somebody walking through a grassy field, that's a living person, and they might squish the plants under their feet. So that would be a biotic factor. Okay, we're talking about earthworms though. So what abiotic things could affect the earthworms? So the amount of oxygen in the soil, they live in the earth, but amount of oxygen in soil. I might want to say oxygen concentration and make that a little bit more fancy in soil. Okay, so different amounts of soil, I guess clay soil, sandy soil will hold different amounts of oxygen. What about pH of the soil? probably pH of soil. They might like acid or non-acid conditions. So that is going to be a biotic factor. The amount of water. So it says they have moist skin. They like water, right? So they've moist skin. So let's say water level in soil or earth. They feed on dead and decaying plant matter. Now that is a living factor. So exchange gases through their skin. I guess that's the oxygen concentration. So we've kind of covered abiotic. I put three things here. You only need to put two things. Let's do the living thing. So they mention there they feed on dead and decaying matter. So I want to say the amount of decaying matter in soil. I guess that's the amount of living things that have died in the soil. Um, Predators. So what eats worms? Birds. I guess predators. You're looking at things like birds that come and pull worms out the soil. So predators in the area. So the amount of predators in the area, the number of predators, and then you've got diseases. Specific to the earthworm. You don't need to write that, but you get what I mean. The earthworm's version of COVID or whatever that's in the soil. So if they're living in an area where there's a new disease or some new predators have moved in, that's going to be a problem. And then of course the amount of food that's available, those are living things. All right, let's zing on, guys. Okay. Students investigated the populations of earthworms in the soil in two different areas. Area A is a grass lawn. Area B is a farmer's field. Chemical X can be mixed with water and poured onto the soil. So we've got kind of chemical X going on. And that can be water. Check out the GCSE level art. Boom. Chemical X. That can be dropped onto the soil. Okay, the mixture brings earthworms to the surface of the soil, but does not harm them. Plan an investigation using chemical X to compare the number of earthworms per meter squared in areas A and B. So what we're going to need to do is, I would say, we want to water area A. So here is area A, and we want to water area B with this chemical X. Why don't you pause the video, write down what you think can get you 6 points. I would say talk about the independent variable, dependent variable, and control variables. And here we go. Right, I'm going to go through the answers now. So you're going to water A and B with chemical X. So water area A and B. And what we want to do is say with the same volume, because it's a liquid, um, and concentration, the same volume and concentration of chemical X. It's like when you make up squash or Ribena or Vimto, right? And you add water, you want to make them to the same strength. So it's a fair test. So each one gets the exact same amount and the same strength, the same concentration. So that's going to get us a mark. Now what we want to do, this might be a big field, it might be 100 meters square. Imagine that this field is 10 meters by 10 meters. You don't want to trudge all across the whole field looking for counting all the earthworms and then dividing by 100, because we need to do it in meters squared. So you could do the whole field, 100 meters, count them all and see if you can count them all, but that wouldn't be very accurate. Much better to place one of those metal squares. Hopefully you remember the name of them. They're called quadrats. Place one of those metal squares. Now, it's easier to say we're going to use a 1 meter squared quadrat. I mean, that's quite big, but it just makes the maths easier, doesn't it? So we're going to say randomly as well. We want to randomly place a 1 meter square quadrat in the field. Now you could explain if you want how you would place it randomly, close your eyes, fling it out, or you could just say draw a grid. So if you want to know the extra details, draw a grid of field on paper, randomly number grid, and then randomly generate a number. You know, on your scientific calculator, you have, you could say using calculator. Now, using your scientific calculator. Now, you can do that by pulling out pieces of paper out of a hat with numbers written on them. That is random as well. Whatever. That's how you decide where you're going to place the quadrat randomly. So that's going to get you a mark. Then what we want to do is say, you don't want to count up the beetles. You need to identify the earthworms. Personally, I'd use pictures on mobile phone. Now, you could just say use something called a key to identify earthworms, because you want to get the right thing, right? You want to check the population, identify earthworms, use pictures on the mobile phone to do so, count number of worms in quadrat, then repeat, I don't know, 5 times. We normally go 10 times with quadrats. Oh my gosh. But 5 is your minimum generally in GCSE science for repeats. So worms in the quadrat, look, I can't spell. Oh my days. Then repeat 5 times and find the mean. And you can explain how you do the mean if you want, but I think the examiner will be cool with that. So you're going to find the mean. So we've said how we're going to identify them. We've said we're going to repeat 5 times and find the mean. And then what we're going to do is repeat the experiment with field B and compare the results to find which has the most worms per meter. Now, remember, we used a 1 meter quadrat. Now, if in an exam they gave you a 0.25 meter squared quadrat, you're going to have to multiply that up by 4 to get it, right? Now, there are other versions of this experiment where they actually say, how would you estimate the entire population? That's not what we're doing. We're finding out the mean population in 1 meter in each field and comparing them. We're finding the mean population. There are other versions of this where you would estimate the whole population living in the field. And in that case, you would do the area. So 10 times 10 is that would make this 100 meters squared. And once you have found the population in 1 meter by doing 5 of these and dividing by adding the total up and dividing by 5, you'd then multiply up to the size of the field, i.e. times 100. So if you had 5 worms was the mean number per quadrat, you would times by the whole field and say, right, there's 500 worms in the field. That is a really common one for them to do, which is estimating the populations in a field. And this is the core practical for B2, the most common core practical that comes up. So make sure you know how to do your quadrat practical. Just checking what else we needed to talk about, if anything else. The only other thing I would say that I haven't mentioned here, I would say this would get you all fat 6 marks. I would say that we need to leave the chemical on each field for the same amount of time before quadrating so that one field doesn't have chemical X for longer, or, the worms don't have chance to go back under because we've left it too long. So leave chemical X on the soil for the same amount of time before quadrating each field. Look, quadrating. How the hell do you spell quadrating? I'm going to say sampling, right? Because that's what we're doing when we use a quadrat, because I can't spell quadrating. So leave chemical X on the soil for the same amount of time before sampling on each field. And anyway, this would have already got me 6 marks because you probably get 1 mark for saying keep the conc the same, 1 mark for keep the volume the same, 1 mark for saying randomly, 1 mark for explaining how to randomly generate, which I put in brackets there. So you probably get a mark there as well. And then 1 mark for saying repeat 5 times, 1 mark for then saying calculate a mean. It's always good to, which I haven't put in there, but to say remove anomalies as well before calculating the mean. Anyway, let's move on. Excuse any spelling mistakes. We're just trying to bang through this. Question 3. Question 3. It's important to control the concentration of glucose in the blood. Figure 2 shows how the concentration of glucose in the blood of a person changed over 4 hours. So here we've got the blood glucose concentration on this graph. We call this the y-axis, and along the bottom here we have time. This is the x-axis. We can see the person's blood glucose shot up there, hasn't it, at a certain time, and then come down, dipped a little there, then back up to roughly near its normal amount. Give one time when the concentration of insulin in the person's blood would be high. Use figure 2, and you need to quote the time in hours there. Why don't you press pause on the video and have a go? All right, I'm going to go through the answers. So when would we expect insulin to be high? Well, insulin is how we lower our blood sugar back to normal. So this is where the sugar is very high, or blood glucose. Glucose is a type of sugar. So this is where it is very high. So this is where we would expect the insulin to also be high. So we want to quote any range in there. I think anywhere between 1.5 and 2.5 hours, and you'd be doing pretty well. So I'm going to put 1.5, but I would imagine on the mark scheme there is a range between 1.5 and, to be honest, probably 2 hours. The insulin is going to be higher than normal between those times. So I'm going to give it 1.5 hours. I'm going to give myself a lovely mark. Why wouldn't I? I'm marking my own test. So there we go. Let's move on. Oh, I'm ticking randomly. You Burke. Let's move down. Okay, 3.2. I love how they use just a couple of lines on a whole page. These people don't think about the trees they're chopping down sometimes, do they? Right, never mind. 3.2. Explain the effect a high concentration of insulin has on blood glucose concentration. It's 3 marks, so we want the effect. What does insulin do to blood glucose, and explain the effect? Right, I'm going to take us up to the graph, but you might want to pause the video and have a go. You should know this off by heart, what insulin does to glucose, the glucose level. What we can see up here, I told you that the insulin, or you worked out the insulin was at its highest around here. What's the effect after the insulin gets high? Blood glucose decreases. And we're going to explain why that is. I'm going to sneeze. I think this ain't... Okay, just had a sneezing attack. I have finished my sneezing attack. So it decreases blood glucose concentration. You can probably just write the word decreases because it's told us it's about the blood glucose concentration. So decreases, 1 mark. Now the explanation. What does insulin do? So insulin It essentially makes the liver cells more permeable to glucose. It allows glucose. So if I want, it makes liver cells. So it's a hormone, insulin. It's made of protein. It has a specific shape, and a little bit like an enzyme binds with its substrate lock and key. The hormone binds to special receptors that are found on the cell membrane of liver cells, allowing the glucose to move into the liver cells by diffusion. So insulin makes liver cells take up glucose. Okay, it actually increases their permeability to glucose, allows the glucose to come in through special gated proteins. But we're just going to say that the insulin gets taken in to the liver by glucose. We could also, insulin makes muscle cells take in glucose. So obviously, if the glucose has moved into the muscles and the liver, then the amount of glucose in the blood will be lower. So I could say the amount of glucose in the blood is therefore lower. So it's taking the glucose out of the blood and putting it into the muscles and the liver. So insulin makes liver cells take up glucose. And what I'm going to just add here is from the blood. And that's the same thing from that there. Now, you probably just need one of those and then to say that that result. So the liver takes in glucose from the blood, therefore there's less glucose in the blood. Now, another thing that it does, insulin basically stimulates or activates, however you want to say it. Basically, it triggers the conversion of glucose into glycogen in liver and muscles. So what happens is glucose is a monomer, a single unit of sugar, and it joins together to make a polymer, a really long chain of sugar molecules called glycogen. So the glucose doesn't even exist anymore, but it's been taken out of the blood anyway. So yeah, you could put that as an extra point there. And let's move on. Right, we're on 3.3. People with diabetes have difficulty controlling the concentration of glucose in their blood. Type 2 diabetes is linked to being obese, being overweight, obesity. Figure 3 shows how to find an adult's body mass. Let's read that again. Whoop, rewind. Figure 3 shows how to find if an adult's body mass is healthy for their height. So we've got the height in meters on our y-axis and then the body mass in kilograms on the x-axis. And then you track that up with your height and your weight and you find out if you are obese, overweight, healthy or underweight. Oh, well, that's scary. I don't want to do that for myself, but never mind. 3.3, person A is 1.75 meters tall in height and has a body mass of 52. Which is person A's category? So remember that 1.75 and 52. I'm going to go up here. I'll write them down up here actually so that you can do that now and you can pause the video and have a go yourself. So person A is, oh my gosh, he's going to forget it, 1.75 meters tall and 52 kilograms of mass. What category are they? Hopefully I didn't make any spelling mistakes, but it could well be. Why don't you pause the video and have a go? All right, I'm going to go through the explanation. So 1.75, we're looking about there for their height, folks. And then 52, we're looking what about? Oh, yes, 55 is about there. So 52 is about there. Now, if I draw a line up, this should be a straight line, but you know, beggars can't be choosers and all that. So it ends about here. And that means when we grid reference that on the graph, they are underweight. So that is the body category that they are in. Let's go down and do the ticky tick on the box. So they would be underweight, and that would score us a mark. Wunderbar. Wunderbar. 3.4, person B is 1.9 meters in height. Give the range of body masses that would put person B in the healthy weight category. So if you are 1.9 meters tall, you're very lucky because that's a very tall height. That's 6 foot something, or around about that. Yeah, 6 foot something. So if you're 1.9 meters in height, what would be the range that your mass can be to stay healthy? Let's go check it out on the graph. So, and why don't you, of course, you can pause the video and have a go before I go over the answer. So we're just going to type that out here for you. Person B is 1.9 meter tall. What is the range? That means lowest to highest of body mass for them to be healthy. Range is always the smallest data number and the highest one. So if you are 1.9, let's get the straight line out. That is where you can first be healthy when we hit that healthy and we draw down there. So what do you think that is? Oh my gosh, is that straight? That might be straight-ish. So about there. And then what is the maximum weight they can be to be healthy? They're still healthy as long as they are there. So ooh, is that 90 or is that closer to 89? I don't know. But it looks to me, they're going to give us hopefully a range here. So it looks like it's between this number and this number. That looks like bang on 90. I'm going to go 90 as the big number. And then they could be 65, 66, 67 to 90 or 68 to 90, something like that. So 67 kilograms to 90 kilograms, something around there would be the range. I'm sure they will allow us one either side. It might be 68 kilos to 90. I haven't zoomed in on that, but there we go. That's going to get you 2 fat marks. Let's zing down. No, it's only 1 mark. Scratch that, but it's 1 mark, but there we go. 3.5, person C is obese. A doctor thinks person C has type 2 diabetes. The doctor tests a sample of the blood from person C. Table 2 shows the results of the blood test. The mean results for people who do not have diabetes. This is one of them confusing ones, going to confuse a lot of people, including myself. Concentration in the blood of person C, they've got a 6.21 cholesterol, they've got a 9.56 glucose, and their insulin is 24.32. Mean for people who don't have diabetes, their cholesterol is only 5.2 and their glucose is half of this person's, it's 4.51 or less than half, and their insulin therefore is less. Type 2 diabetes occurs when body cells have a reduced response to insulin. Give 2 ways the results of the blood test show the person C might have type 2 diabetes. Well, look, what did we learn about insulin? We learned that insulin should reduce glucose. They've got high insulin. Has the glucose gone down? No, but this person doesn't have as much insulin. Their glucose is way down. So what I'm going to say is they have a high insulin concentration. I've longed this out, but high blood insulin concentration, that's the first point. And then you might say, should we comment on cholesterol? Cholesterol's not really linked to diabetes. Cholesterol, folks, is more linked to heart disease, which you do in paper 1 in the organization topic. So we're going to go with the high amount of glucose as our second point. So they have a high blood glucose concentration. And that is going to get us another mark. There we go. All right, let's zing on and see what else is in store in this question. We're right at the end of this question. I couldn't come quick enough. Give 2 ways that a person can reduce the chance of developing type 2 diabetes. So type 2 diabetes is weird. You could say, oh, take insulin injections, but that would be wrong. Type 2 diabetes is you still make insulin. You still have insulin to reduce your blood sugar. The problem is your liver cells and your muscle cells, the cell membranes of them no longer respond. They've become resistant. They no longer listen to the insulin and they don't open their little glucose gates and let the glucose in. So they've stopped listening. So it's not that the glucose is there. We use the biological term resistant. The liver and the muscle cells have become resistant to the insulin. They don't listen to it anymore. So giving them insulin injections is not going to work because their cells are not listening. So the thing you need to do is lower their sugar level. Can you think of how you could lower the amount of sugar in someone's blood? Of course, we could restrict carbohydrates. Carbohydrates, rice, starch, pasta, things that are high in starch like rice and pasta and bread. Those break down to form lots of glucose. So restrict carbohydrates and sugars in diet. I reckon you could get either of those. If you said a low sugar diet or a low carbohydrate diet, that is going to give you a mark. Gosh. And the other thing that can use up glucose is to make your body do more respiration. Now, how can you make your muscles respire more? You can do more exercise. Do more exercise, or a better thing would be to say increase levels of exercise. And there we go. Okay. Someone with type 1 diabetes might want to do that as well. Those two are valid for type 1 as well. Question 4. The rapid growth in the human population means that more waste substances are released into the environment. The release of substances into the environment can cause pollution. Name one harmful substance that can cause air pollution. There are 3 of these you need to know. So there are 3 possible answers on the AQA biology specification. Why don't you press pause on the video, rack your brain, see if you can do it? Okay, I'm going to go through the answers. So there are 3 possible ones. We have nitrogen oxides. That can be dioxide as in NO2, NO3, or NO. We have carbon monoxide. That can be the one that joins to your hemoglobin and restricts the amount of oxygen carried around your body. And then we have sulfur dioxide, which can cause some respiratory problems. It's a choking gas, but also it causes acid rain. And so does NO2 cause acid rain. All 3 of these are harmful air pollutions, guys. So you can use any of those. One that I would say, beware, major beware, big misconception. Students will mess this up. Slap yourself in the face if you wrote carbon dioxide. Carbon dioxide, we're told about how evil it is, but it's not inherently a harmful substance to humans and to life. In fact, without carbon dioxide, life on Earth wouldn't exist in its current form. Carbon dioxide is a very important gas, so it's not a harmful air pollution. It is a global warming gas, a greenhouse gas. So do not put carbon dioxide down as a type of pollution. It's indirectly harmful to us in large amounts, but you're breathing out carbon. Every mouth of air, every bit of air you breathe in contains carbon dioxide, and it's not harming you. So, yeah, don't write carbon dioxide. Name 3 harmful substances that could cause water pollution. Do not refer to plastic or litter in your answer, and it's worth 3 marks. 3 harmful things that could end up in the water and cause water pollution without us saying plastic. Why don't you pause, have a go? All right, I'm going to go through. You probably heard of like crude oil from your C2 paper. So crude oil is the thick black mixture that the world runs on, essentially. Yeah, but when that gets in the sea, it gets into the feathers of birds and seals and fish gills and kills them. So when there's an oil leak from a tanker or a whatever, an oil rig. Now, so crude oil is one possible answer. Another thing that could get is their chemicals from a factory. So some industrial places dump their chemicals naughty into water, and that can be a problem. We could name some if we want. Heavy metals, things like mercury, stuff like that would be bad. Other things would be fertilizers. Now, farmers all over the world use fertilizers to make their foods grow better so they can feed the population, get a higher yield of food. Unfortunately, when it rains, these leach into the sea and they cause algal blooms, etc. So fertilizers are a problem. And of course, when it rains heavily, sewage systems overflow and contaminate the water. Sewage, like poo, basically feces, urine, and that stuff can get into the water and be a harmful substance. So sewage is another one there. There's possibly other ones. You'd have to consult the mark scheme. Check out the link in the description for that. Right. Let's zoom. Oh, it's another six marker. Oh, we love it. Not. 4.3. Describe how substances that pollute the air and water could be harmful to humans and other living organisms. Right. They've set us up for this by us talking quite a bit about stuff. So that's great. We've got loads of information earlier in this question that could get us through here. Look, we've already gone through the water. So now explain how those three things we just mentioned in the water could be harmful to humans or other living organisms, and talk about how the air pollutants could be harmful. It's worth 6 marks. Grab a pen, grab some paper, scribble it down, have a go. You'll easily smash out the marks here. I would go for 3 for the air, 3 for water. But to be honest with you, probably 2 of those and 1 of those, or 2 and 2 with the explanation, is going to be good enough for the fat 6 marks. Why don't you pause the video and have a go? Okay, I'm going to go through the air pollutant factors first. So I'm going to say nitrogen oxides. These are nitrogen oxides from car exhausts. So these actually come out when, I don't think you'll need to put this, but when nitrogen and oxygen, oh my gosh, I'm explaining how they formed, react on hot car engines. Obviously, car engines are made of metal. They get very hot and they kind of act as like a catalyst to join the nitrogen in the air to the oxygen. You get this reaction where you get N2 plus O2 and it forms like NO2 like that. And if I want, I could put that there and put a 2 there, balance that up, but who cares? Essentially, the key here is nitrogen oxides. And what do they cause? What is their big issue? They cause acid rain, which changes pH of soil, kills plants. Okay, so it also does plenty of other things, but I'm just going to leave it at that. That's talking about how it harms other organisms. So that's some of the stuff it does. You're going to get at least 2 marks for that. Also, nitrogen oxides are nitrogen oxides. They can cause respiratory issues, I guess, in humans, things like asthma. So if there's a high level of nitrogen oxides in the air, it's going to cause things like asthma as well. So that's something you want to talk about. Another one that is from the air is sulfur dioxide. That's SO2 in its formula, causes acid rain. I don't think we need to explain how it does that, but essentially it dissolves in the water in clouds to form sulfuric acid or dilute sulfuric acid, and it waters down again, changes pH of water sources, and kills fish. So it changes the pH of the water, makes it more acid, it kills fish. And so does nitrogen dioxide. They both do it, but I thought I've already mentioned the fact that it can change soil pH and kill plants and trees. So I'm just going to mention that the sulfur dioxide, which is also acid rain, kills fish. It can also cause respiratory issues as well because sulfur dioxide are choking fumes. So choking fumes cause respiratory illnesses. So sulfur dioxide can cause respiratory illnesses, okay, things like asthma, breathing problems. Now we've got a third one. Do you need to put all of this? No, but like I'm just going to bash it all down so you can get like all of it. And in an exam, if I just came out with 2 of these, I'd be perfectly happy. The third one is carbon monoxide. Ooh, carbon. Oh my gosh, I'm having issues. Carbon monoxide. This, yeah, what am I going to say? Poisons hemoglobin, prevents oxygen transport in blood, so toxic to humans, can be fatal. There's a lot I could waffle on about it. You're going to feel tired, less oxygen, less aerobic respiration in organisms, and so you're going to be tired. It binds to the hemoglobin and prevents oxygen being carried in your red blood cells by hemoglobin. I could go on, and we could add, like, where did we stop? But that's going to be more marks. So what I'm saying is, I think, like, if you just talked about air, you probably restrict yourself to only 4 marks. Even if you bust out all of this wicked information, you'll probably only get 4 marks. So because they've said air and water, we need to talk about water pollution as well. Now, if you did 2 of these, that would be enough for air. So for water pollution, I'm going to say there's loads of things we could do here. Fertilizers pollute water. They cause algal blooms, block sunlight, prevent photosynthesis. Some of you might have heard of eutrophication, which is the process by which the algae blocks the sunlight. They prevent photosynthesis, kill plants in the water, and deoxygenate the water. There's more than that. The bacteria feed on the dead plants when they die and use up all the oxygen in the water, and then that kills the fish. Okay, so I don't want to waffle too much, but that is one of the ways that fertilizers can smash up the water. Another thing, we could just say plastic, fishing nets, plastic fishing nets in water. Get stuck in gills of fish, turtles. So basically, they mash up the sea organisms, seals, turtles, things that live in the water. Plastics are smashing them up, okay, and making them very ill. So they get stuck in their gills, stuck in their lungs, stuck on them, all of that kind of stuff. So plastics in the water, bad, bad, bad. That's another type of pollution. What else we can say? Oil spills. the crude oil sticks to feathers of marine birds. Marine, what do I mean? I don't mean like they're in the Marines. Hoo-ha, soldier. I don't mean that. I mean in the Marines as in marine meaning the sea. Yeah, so they stick to the seabirds that like to land on the sea and eat fish. The oil gets into their feathers, and of course that means they can't fly and they drown and they die. So that's bad, bad, bad business. We talked about sewage, feces. Oh my gosh, feces, urine, etc. Chemicals, toxic from factories, kill fish. Let's just write kill fish a lot, right? So sewage, chemicals from factories, kill fish. You get the idea. There's loads and loads of things we could go on. I could go on and on and on. We're just going to mention a few of them in the water. We've got fertilizers, we've got plastics. We talked about the oils. Then there's sewage from sewage plants and then chemicals from factories that leak into the water. So there's tons there to get the marks. If you want to look at all the many, many marks, you would have to consult the mark scheme, which you'll find in the link. Question 5. Maple syrup urine disease. Oh my days, maple syrup urine disease. Oh, MSUD is a rare inherited human condition. MSUD is usually diagnosed early in childhood and can be controlled by having a low protein diet. Figure 4 shows the inheritance of MSUD in one family. So we've got males with MSUD, they're these coloured in squares. Males without MSUD are these white squares. And then we've got females with MSUD, they're these shaded or coloured in circles or black grey circles. And then females without MSUD, they're these like white circles here. The allele for MSUD is recessive, so that's a key thing you should underline or highlight when reading the stem of a question. Give one piece of evidence from the figure 4 that shows that MSUD is a recessive condition. So these are quite hard. These are called pedigree diagrams. You have to be a bit of a detective to work these out. What is one piece of evidence that is going to tell you that it's a recessive and not a dominant condition? And what I suggest you do is you look at where two parents, i.e. 3 and 4 or 1 and 2, have had children. And if you can see evidence that proves it's a recessive condition and not dominant. All right, pause the video, have a go. Right, I'm going to go through the answers. If you look at 1 and 2, let's just pick on these two, for instance. Both of these, we've got a male without and a female without, 1 and 2, and they've had a child with. Now, how can they not have the disease? So let's say we use T for the disease and we say, right, it's recessive. The only way they can have their child can have the disease is if it's recessive. If it was a dominant disease, if it was found on the capital T, then whatchamacallit, both of these parents would have the disease. So the only way they can have, the only way that the parents can not have the disease is if they are carriers. God, my brain froze there for 2 seconds. If you're wondering what the pause was, it wasn't a glitch. So parents are carriers. You can't have carriers with a dominant a disease caused by a dominant allele. So as I say, if it was on the capital T, then both these parents would have it. The only way they could not have the disease if they were little t, and then all of their children wouldn't have the disease. So if it's a dominant disease and you have two parents without the disease, then all of their children wouldn't have the disease. To have two parents without the disease and one of their children has the disease, it must be recessive because the parents are carriers. So we can say parent 1 and 2 must be carriers or heterozygous, as one of their offspring, 5, has the disease. Okay, there we go. There's our evidence. Let's move on. Don't beat yourself up if you find these hard. They are difficult, these pedigree diagrams. Right. Persons 7 and 8 in figure 4 are expecting a 4th child. Determine the probability that the child will have MSUD. You should draw a Punnett square diagram. Identify the parents. That's going to get you a mark, the phenotype of each, and identify the phenotype of each offspring and the genotype. Use the symbols R. We're being told to use capital N and little n. Little n is the allele for the disease. Big N is the allele for without it. We're looking at parents 7 and 8. So I'm going to go up and use this, and then we're going to show the probability. What did it ask us? The probability that, determine the probability the child will have MSUD. Okay, so 7 and 8 are having another child. So we're going to look at 7 and 8 and check out what they're, have a look at 7 and 8. They do not have the disease, 7 and 8. And we know that they must be carriers. They must be big N, little n. How do we know that? Well, look, they've got a child who was NN and had the disease. It told us the disease is recessive, so you must have little n to have the disease. We just need to do the rest. So let's smash out a Punnett square and I'll show you how you get the marks. When I do a Punnett square, I make a noughts and crosses board like that, and then I shade out this because I'm not interested in that. And then that could be mum, and this could be dad. You can use a symbol for that if you like. I can't remember the symbol, but so mum is going to be big N, little n, and dad is going to be big N, little n. So they're going to give you one of the 4 marks for identifying what we call the genotype, the type of alleles that the parent has. Now we're going to go and work out the genotype of the offspring. So we've got big N, big N. I'm doing this big N times that little n. We get big N, little n, then little n times big N, but you always write the big one first. So again, it's another little n and big N together. And then we have little n going with little n. Now, those genotypes, the correct genotypes that you've done there is going to get you another of the 4 marks. Now, what we need to do is say what each of these is. You can, if you've done this big enough and I haven't, you can write it in here. So I'm going to write it in here. This person, so who has the disease? Only one of these has the disease. So I'm going to say MSUD. That is the phenotype. Everyone else, no, I'm going to say no MSUD because I can't fit without no MSUD. Oh gosh, let's not do that. That's looking ugly. I'm going to do a little key for the examiner here, and you could do this in your exam, but I'm going to basically say I want the examiner to know that I know that these have no MSUD. You might say, what am I doing by writing this? This is the phenotype. So the way that the genes are expressed, they don't have the disease. This person does have the disease. So I'm showing the examiner I know what the phenotypes are. That's another mark. And now so that's 3 marks. One for the phenotypes, the MSUD and the no MSUD, one for the genotypes, one for the genotypes of the parents. The 4th mark is to work out the probability. Well, it's 1 in 1, 2, 3, 4. So 1 in 4 is the probability. So probability is 1 in 4. You can put 0.25. You can put 25%. They will all be worth that 4th mark. Now, I have seen 6 markers where they say determine the probability that the child is also a girl or also a boy. So if we said you had to work out the gender of the child as well. So they're NN, MSUD, and it's 1 in 4. Well, there's a 1 in 2. So you do 1 in 4 times 1 in 2. So 25% times 50%, and you get 12.5%, or like 1/16. So this is 1 in 4, isn't it? Or 1/8, sorry. You get 1/8 or 12.5%. Okay, let's zing on because we don't have to work out their gender, but I'm just making you aware that some exam questions on this have been 6 marks and have included gender. Okay, here's the next question. Figure 5 shows chemical reactions involved in the normal breakdown of some types of amino acid inside body cells. So we've got figure 5, some amino acids. There's an enzyme creates a toxic substance P or creates ammonia. And then other enzymes will turn ammonia into urea and toxic substance P, another enzyme. Enzyme 2 turns it into a harmless product that will not damage the body. A person with MSUD cannot make enzyme 2. So they get toxic substance P. One of the final products shown in figure 5 is urea. Where in the human body are the reactions shown in figure 5 most likely to occur? So where does urea get made? Now, the common, pause the video, have a go. Or I'm going to go through the explanation. The common mistake will be people will say, oh, it's made in the kidney. No, the kidney gets rid of urea. We wee it out when we make our urine, but that's not where urea gets made. The correct answer is the liver. This is where we deal with toxic chemicals, toxic substances, lactic acid that we get from anaerobic respiration. This is where we deal with ammonia. This is where we deal with harmful substances, is the liver. Okay, enough whiffle, waffle, waffle. Let's move on. Scientists can analyze samples of urine. Let's rewind. Scientists can analyze blood samples or urine samples to see if a person has MSUD. The test identifies high concentrations of toxic substance P. Shown in figure 5, explain why the blood of a person with MSUD will have a high concentration of toxic substance P. Use the information from figure 5. It's worth 3 marks. This is really easy, this one. It tells you that, look, normal people break amino acids, get turned into toxic substance P. However, people with MSUD cannot make the enzyme 2, so they can't break down toxic substance P into harmless products. So if you have a lot of toxic substance P in your body, that is because you do not have enzyme 2, so you cannot break down you can't do the lock and key and break down the toxic substance P. So explain why the blood of a person with MSUD will have a high concentration of toxic substance P. So they do not have enzyme 2. Enzyme 2 breaks down toxic substance P into harmless products. So they still have toxic substance P in their blood. So essentially, it's not being broken down, and that is what we'd say. It's kind of like the substrate that's broken down by the enzyme. Okay, explain why the urine of a person with MSUD will have a high concentration of toxic substance P. So this will be that it will get filtered out in the kidneys and where it will be turned into urine. So there we go. Toxic, oh my gosh, I can't type, I'm losing the ability. Toxic substance P will be formed into, or let's say will be filtered. We don't, when we're talking about the kidney, we want to use the word filtered, will be filtered by the kidney out of the blood and is not reabsorbed. You have something called selective reabsorption. Toxic substance P is not going to be reabsorbed. So it is not reabsorbed. It is excreted in the urine. So there we go. Okay. Oh my gosh. Does this question never end? It's 14 marks. What a long question. Explain why a person with MSUD must have a low protein diet worth 3 marks. Let's go and check that diagram in figure 5. And this is how I'll explain. Why is it good for them to have a low protein diet? Look, protein, when we eat protein, it gets turned into amino acids. If you have amino acids, you're going to make toxic substance P and you're not going to be able to break it down. So it's going to harm you because it's toxic. So a low protein diet means less amino acids, less enzyme 1 turning into less toxic substance P. So let's go put that down into our answer. Where are we? So we want to say less protein means less amino acids formed when protein is digested. This means less toxic substance P formed when enzyme 1 converts amino acids to protein, which is good as toxic substances damage cells in the body. So we potentially need to explain why toxic things are bad here, because toxic substances damage cells in the body. So you're going to have less toxic substance, less damage to the body. All right, let's zing on. Oh my days. What are we on? Question 6. Energy flows through an ecosystem and materials are recycled. Figure 6 shows the water cycle. You've got the sun here. You've got something going on there. You've got something there, something there. You've got something coming up through the roots of that plant and out the leaves there. And you've got something coming up from the water into the air and something from the soil into the lake. Okay, name process X. So we've got some sort of water here, like a lake, and it's going up. Why don't you pause and have a go? I'm going to go for the answer. That should be fairly obvious. That is going to be evaporation. So yeah, it's evaporation. Okay, and that will get you the mark. This should be a fairly easy one on the old water cycle. It's basically a quite heavily content-based topic. So name the process by which water is absorbed into plant roots. So you should know this from your paper 1 biology. How does H2O get into the roots of plants? And the answer is, pause the video and have a go, osmosis. So hopefully you know that water enters the roots by osmosis through those root hair cells. Give 2 uses of water in plants. What do plants use water for? So plants use water for photosynthesis. Ooh, photosynthesis. Everybody knows this. So for photosynthesis, it's a raw ingredient for making glucose. So I might want it for photosynthesis to make glucose. It's where you get the 12 H's from in glucose. So they come from the water. So what else can the water be used for? The water can be used to transport mineral ions. So how do they get like Nitrates. Nitrates are a soluble mineral ion that are needed to make amino acids, and they are taken up the xylem. Um, yeah. Also, what I would say, you can transport mineral ions. You can also move glucose and amino acids around in the phloem by translocation. We need water for that. And I would also say that water can be used as support in the xylem or in the stem. It keeps the plant upright. When the cells are turgid, that keeps the plant standing up. So lots of different ones there. Those are the two I went for. Let's move on. Okay, figure 7 shows the flow of energy through a food chain. The numbers are in kilojoules per meter squared per year. Bit complicated that. KJ meter square year. We've got sunlight, 1.7 million going into the grass. Now the grass uses 20, it grows and it's got 21.8 21,800 kilojoules left. 15,200 kilojoules goes to decomposers. 3,550 goes to other herbivores like rabbits or whatever chews on the grass. And then 3,050 of the kilojoules goes into the cow. The cow uses 125 kilojoules for growing and releases 1025 by respiration. So it's like heat to the environment or carbon dioxide from its mouth. And it poos out feces, excretes 1900. All right, let's zing down. They're going to give us a maths question. I'm taking it. Yeah, baby. 5 marker. The cow is more efficient than the grass at converting energy. The energy conversion efficiency of the cow is 4.098%. Calculate how many more times efficient the cow is at converting energy than the grass. The equation for energy conversion, they've been very nice here, is energy used for growth divided by energy input times 100. Give your answer to 3 significant figures. So we've been given how good the cow is. They've told us that the cow is more efficient, which means our answer should be less than 4.098%. Why don't you find out first of all? So we know that this is 4.0. Oh my gosh, was it 98? I believe percent. We'll double check that in a second. We need to find out what percentage of efficiency the grass has. Look at the total going in. Look at the total amount that goes into the body of the grass. Why don't you calculate that and then find out how many times more efficient the cow is than the grass at transferring energy? Why don't you pause the video, have a go? All right, I'm going to show you how to do this. So this is the amount of energy that went into the cow for growing, into the grass for growing new leaves, roots, etc. And the total amount of energy that went into the grass in totally is 1.7 million. So I want to do 21,800 divided by 1,700,000. And then I want to times that by 100 and work that out as a percentage. Okay, they are 1.28% efficient. So the grass is way less efficient, right? And we were told that. How many times less efficient is that? So I would say you're probably going to get yourselves 2 marks for that out of 5. Now what we need to do is go 4.098, and we're going to divide that by 1.28, and that is going to get us another mark. Let's work out how many times more efficient it is. So let's do this. 4.098. I hope it was 4.098. I'll have to go down and have a look. And that is 3.20156. So it's 3.20. Oh my gosh, 156 times more efficient. I guess that's going to get me 1 more mark. And then they asked for 3SF. I'm notoriously bad at these things. So 3 significant figures. That would get us 3.20 as the correct answer. You start from the left and you do the first 3 solid numbers. Now, the first 3 solid numbers are 1 there, but it's not enough to push that up to 3.21. If it was 3.205, then it would be 3.21. Now, I just want to double check my figures because I could have made a mistake. 21,800. Let me just do that again over there because I may have not done that to the correct number over there. 21,800. I want to divide that by 1.7 million. And that gives me, oh, right. Actually, that gives me, let me zing this. I need to do this a little bit more carefully. That gives me 0.01282. I got a bit crude here, and I think this may change my answer down here. I'm going to times that by 100. And that's given me 1.2829. So equals 1.2829. 4-1, blah, 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 blah. And so if I change that here, and I add 29 there, how does that change my answer there? Let's go ahead and do that and see if it changes my answer at all. Okay, sorry about this, folks. 4.098 divided by 1.2829. It's actually 28941, but I can't be bothered to do all that. Right, I've got 3 point, that has given me a slightly different answer. So that has given me 3.194. I think if I kept this number whole and put 41 there, that would be like 3.195 or 96, but it would be the same thing. Let's see. Oh, I'm just going to write the whole thing in as I think that was 4-1. Let's try that again and see. I think I've been a little bit chiefed and actually it is. I need to be a little bit more accurate. What can I say, folks? Sometimes, you know what? I'm not as accurate as I should be. Um, oh my days. 1.282352941. Oh my gosh. 1.282. 1.282. Oh, disgusting. Oh God, I'm going to write it out here. 1.282352941. That's painful. So let's see what that equals. 1.282352941. Look, I'm sure they'd have accepted my first answer. I don't know why I've got carried away doing this. This happens to me sometimes. I get myself in a right old twister. Right, 1.282352941. I'm starting to lose the will to live. Right. Hard of my days. So if you manage to keep all of that fat one, no, did it in your calculator, then you'll get this 3.1956. And after all that effort, it doesn't make any difference. I still go there. So even if I use 1.28, it works out the same anyway. What an idiot. Right. That is larger than 5, basically 5 or above. We're going to round that to 3.20. So the answer does not change. Even if I do it to super high accuracy, you still end up with 3.20 to 3SF. I've just wasted 5 minutes of everybody's time. Sorry about that, folks. Okay. All right, we're on 6.5. It is more energy efficient to rear cows indoors than to rear cows outdoors. Give 2 reasons why. So, why is it more energy efficient to rear cows indoors than outdoors? So, one of the reasons is think about when you're outdoors, what happens to your body right now? It's about -5 in the UK. You're going to be shivering, and that means you're going to be losing heat to the surroundings. So, indoors is warmer. Indoors is warmer, so less heat loss to surroundings. Um, so more mass gained by cow. So if you keep your cows indoors in a cow shed, they're going to grow bigger than if they're in a field. Hence why your field organic cows are more expensive because the farmer has to account for the fact that they've got to feed them more to keep the cows warm because they're losing heat to the surroundings. Another reason why cows in outdoors is cows move around less indoors. So indoors, cows cannot move around as much. So less energy transferred to like kinetic energy, more energy for, whoa, for mass growth in cows. So your cows are going to get bigger if they're indoors because they can't move around about as much. A bit like if you sit on the sofa and you play on the Xbox all day, you're not moving around as much, you're going to get bigger. And that's my issue. Right. 6.6 suggests 2 possible disadvantages of rearing cows indoors. What if you're really close to other organisms? What can pass between you really easily? So what we're going to say is diseases spread more easily. Again, you could pause the video and have a go at these before I write them down. Diseases spread more easily as close contact. Um, let's have a little thing. Less healthy for cows. So less healthy for cows as maybe they get less sunlight on their skin. Sunlight's good for vitamin D. It must be good for cows as well. Less freedom to roam, maybe less ethical. You know, if you could give cows a choice, maybe they'd say they like to be outside and roaming about free with a lot of space. So less freedom to roam, less ethical, less healthy for cows. There's less sunlight. I guess their excretions, they can't get away from their poo. They're going to be standing in each other's excretions and stuff like that, which might not be too healthy as well. All right, guys. Okay, question 7. A scientist found a polluted pond which had a new type of blue algae in the water. The blue color of the algae was caused by a mutation. What is a mutation? So, why don't you pause the video, have a go? All right, I'm going to go through the answers. So, what is a mutation? A random change in a gene. That's your GCSE explanation. If you prefer, you can say a change in the nucleotide base sequence or a change in the base sequence of a gene. Of course, there we go. A gene codes for a protein. Hopefully, they won't ask us that next. Otherwise, I'll just give them the answer away. Anyway, a random change in the sequence, base sequence for a gene. Right, let's move on. You can just say a change in a gene. Okay, the scientists measured the number of blue algal cells in a sample of the pond water. The scientists used a special slide which has a counting grid. This is the method used. Dilute 2.5 centimeter of pond water up to a volume of 10 centimeters by using distilled water. Place a drop of the diluted pond water on the special slide. Now, we can think about this. That's 2.5 and it's gone up to 10. So they've kind of diluted it 4 times. They've made it 4 times weaker. Place a drop of the diluted pond water on the special slide over here. Place a thick cover slip. There's the thick cover slip over the water. To give a depth of 0.1 millimeter of water. So we've got a depth, it's 0.1 millimeter. Use a microscope to count the number of algal cells in a 0.2 times 0.2 millimeter square on the counting grid. Okay, let's go down. I think we're going to have some maths here, folks, because they're giving us numbers and data. Figure 9 shows the view of the counting grid through a microscope. So these are the little cells. I think that's their nucleus we can see. And this is the counting grid. And we've got the overall size, 0.2 times 0.2 millimeters. Okay. How many algal cells are in the 0.2 times 0.2 millimeter square in figure 9? Use the following procedure. Count all the cells that are completely within the 0.2 millimeter square in the counting grid. Count cells that are touching to the left side or the lower side of the square. Do not count cells that are touching the right side or the top of the square. How many squares are in there? So why don't you have a go and count how many squares are in the counting grid based on the rules where if they're on the left or the lower of a square, they count. Okay, I'm going to go through and show you the correct answers. So we want to count. We don't count that. That's at the top. Nothing at the top we count. Nothing on the right we count. And that was the rules, basically. Everything else we count if it's touching. These are not touching. This is not touching. This is not touching. But we are going to count that. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. It looks like 16, folks. I think I've got them all. So 16, of course, we didn't count anything on the top or on the right, and only if it was touching the bottom, like this one, or touching the left, then we counted it. But these, unfortunately, were not touching. They were clear gaps, so we didn't count them. So 16, I believe, is the correct answer, and that is going to get you one of the marks. And that's in the 0.2 times 0.2 grid. Okay. 1 week later, the scientists repeated the test and counted 14 cells on the 0.2 counting grid. Calculate the number of algal cells in a 1 millimeter cubed of undiluted pond water. Use the scientist's second count of 14 cells. It's worth 6 marks. This one is a maths one. It's quite difficult. So grab some paper, grab a pen. Why don't you pause the video and have a go at this one? Before you actually have a go, let me give you the data that is necessary. We were told up above that it was 0.1 millimeter deep. So when we're looking at this, it was 0.1 millimeter deep. We know it's 0.2 wide and 0.2 across. So we know how to work out the volume of the counting grid. It says calculate the number of algal cells that would be in a 1 millimeter cubed of undiluted pond water. We also know that this count of 14 cells was in the diluted pond water. So I'm giving you all the information before you have a go. Dilute, so you don't have to go and I don't have to scroll up for you. So that is diluted pond water, which is 4 times weaker. How do I know it's 4 times weaker? Because we went from 2.5 centimeters, remember, we went to 10 centimeters. So cubed. Yeah. So we know that they weakened it by 4 times. So when we work out our answer, we need to multiply it by 4 to get the amount in the undiluted pond water. Anyway, it's worth 6 marks. Why don't you pause the video, have a go, and then unpause, and we'll go through the walkthrough. Okay, let's go through this for you. Now, what they've told us is they found 14 cells in 0.2 times 0.2 on the grid. They found 14 cells in that. But remember that this was also 0.1 deep. It told us that up in the stem of the question. So what is the volume of that? Well, I'm going to do 0.2 times 0.2 times 0.1, and that is going to give me the volume of our little slidey thing, and it equals 0.004 millimeter, because these were all in millimeter to the 3, because look, millimeter times millimeter times millimeter gives me millimeter to the 3. So 0.004 millimeter is our volume of one grid, which contained 14 cells. So how big is how many lots of these grids, how many lots of 14 are in 1 millimeter? So what I want to do is go 1 millimeter cubed. I'm going to divide that by 0.004 and find out how many of these I can fit in 1 millimeter. And the answer is 250 of these slides. Now, each of these slides has 14. So I want to times that by 14 to get the total amount of cells that would be in 1 millimeter cubed of diluted water. And that equals 3,500 algal cells in in 1 millimeter cube, but that would be for the diluted. And they've said they want it in undiluted. Remember, we found it out in the diluted. We need to convert it back and find out, well, how much would be in the original. Well, we need to times it by 4 because they made it 4 times weaker by diluting it. So we're going to times it by 4, and we're going to find that actually there are 14,000 cells once we make it back by timesing it by 4 in the undiluted, the pure strength pond water, 14,000 cells. And that's going to get you 6 bad boy marks. That was actually a really hard question. Difficult maths to do there. Right, let's zing on. Suggest why the scientists diluted the pond water before placing it on the special slide. So how many cells did we have on that slide? Well, the first one we counted was 16. Look how hard it was to work out how many cells. Now imagine there were 4 times the cells. So 4 times 16 would be 40, 4, 6 is 24, 64 cells. How would you actually count them all? It'd be very hard to work out which ones were in and which ones were out. It made it easier to count. So the reason we dilute samples is to make it easier to count. It was easier to count in the diluted sample as well as less cells. Okay, so easier to count, lovely jubbly. Right, 7.5, a student repeated the scientist's method. The student used a thin cover slip over the diluted pond water instead of the thick cover slip. The liquid pulled the thin cover slip downwards slightly. Explain how the use of the thin cover slip would affect the results for the cell count. Ooh. So in the original one, just to show you, you might want to pause the video and have a go. In the original one, they had a kind of dip like that, and they filled that with the pond water with the cells in. Then they put a fat cover slip on. Now, the fat cover slip did not alter the shape of that. But when they've used the thin cover slip, let's have a look what it's going to do. The thin cover slip bends down in. What does it do to the volume in there? It makes the volume smaller, right? It makes that volume smaller because it bends down with the thin cover slip. Because I mean, if you squish this thin cover slip down, you're going to get some water leaking out, right? Leaking out the sides like that. You're going to squish some of the water out. That's why they use the fat cover slip, not to change that shape. So you're definitely going to make the volume smaller, makes the volume smaller as some water leaks out. And I guess that water that leaks out might take cells with it. You lose some cells. So less cells in the cell count. So it's going to appear like we have less cells because some of the water leaks out. We have a smaller volume, less cells, essentially. Lovely. Right. That's the end of that question. Let's get on with question 8. Don't stay up late. It's question 8. Doing the exam, concentrate, whatever. Right. Hey, oh my days. An echidna is a mammal that lives in Australia. Cheese. Echidna. Let's go with echidna. Whatever. Yeah, figure 10 shows one of those. Figure 11 shows how the body temperature of the echidna varies in warm weather and in cold weather. So here's our little hedgehog-like creature called an echidna. Looks kind of cute. How does its body temperature vary in warm and cold weather? Here's its body temp. Now, in the cold weather, we can see its body temperature goes up, down, up, down, up, down. And in warm weather, it seems to stay hotter and up and down like that, time in weeks. This shows how a human body temperature varies. So this is a bit like the echidna in the warm weather. Compare the variation in body temperature of the echidna in warm weather with the variation in body temperature of the human. So I would say they're quite similar, aren't they? Look, theirs varies around 30 degrees, and then it goes up to, what would we say? Now it's a describe, so, or compare. So you're not explaining, you're just saying what you see. So I would say the echidna goes up to, what's that, 35, and at the lowest it's about 27, and up to 35, and it fluctuates within that, up and down sort of twice a week within that. The human body temperature, it varies around 37. It goes to the lowest at 36.1, and the highest is about 37.1. So it's got a 1 degree range between 37.1 and 36.1. The echidna has a larger range. It's going between 27 and 35. So it's a larger 7 degree range, but they both fluctuate in a similar pattern. So I would say similar fluctuations. What is a fluctuation? That's the going up and down. Similar, I can't spell, similar fluctuations. Similar fluctuations. However, the human body temp highest is 37.1 and lowest, I don't know, 36.1. So a 1 degree range. So a 1 degree kind of range from 37.1 to 26. Oh, 36.1, that's supposed to say. Let me just write over that. That's 36.1, supposed to be, folks. Whereas the echnid, I can't even say that one. Whereas the echnida. Oh yeah, let's leave it at that, man. It should be a capital E, whatever. Whereas echnida has a larger range of fluctuations between, I think we said 27. Was it 25? Oh my gosh, I've completely forgot what the range was for that echnida thing. Let's have a little look. What did it go down to? 27 and up to 35. Yeah, so it was like an 8-degree fluctuation. So whereas the Echinidea had a larger range of fluctuations between 27 and 35 degrees, so 8-degree range. Yeah, there we go. Lovely jubbly. That's going to get us the marks. I'll stop ticking. It's all falling apart. I'm tired. In cold winter months, the echidna, I did spell it wrong in my answer. In cold winter months, the echidna hibernates. During hibernation, the echidna's body temperature decreases below 5 degrees. The echidna sleeps for 17 days at a time. The echidna's rate of metabolism slows down. Explain why the decrease in body temperature is an advantage to the echidna during hibernation. So, if it decreases its body temperature, then it is going to loses less energy as heat to the environment. So it loses less energy transferred as heat to the surroundings. So it has more chemical energy, more energy stored as fat, or more energy for insulation and for respiration and metabolism. So you've got these metabolic reactions keeping you alive, keeping your breathing, keeping your heart rate going, all of those things, making cells, growing, repairing, doing all of those jobs. If you're losing less heat to the surroundings, look, humans are at about 37 degrees, 36.4 to 37 degrees average body temperature. And that means if we're out in 0 degree, or if we're out in like a really cold climate where it's 0 degree, there's a 37-degree difference. Heat always moves from hot to cold. So there's going to be a rapid loss of heat from our body to the surroundings. Now imagine with the echidna's body temperature down below 5 degrees, imagine that the cold winter months are 0 degrees, then there's only a 5-degree difference between its body temperature and the surroundings. There's going to be less heat transferred, or it's going to be transferred less quickly to the surroundings, which means it has more energy to keep it alive by doing other important things like respiration and other important metabolic reactions. So less energy lost as heat, more energy for staying alive by doing other metabolic reactions, less food having to be broken down, less glycogen and other molecules in its body, fat and things like that. Okay, cool. Let's move on. 8.3. During hibernation, the echidna wakes up several times. Each time the echidna wakes up, it becomes active and its body temperature increases over 30 degrees. Explain why the echidna has a high body temperature when it is active. So why does its temperature increase and go really high when it is active? Why don't you pause the video? Oh, echidna is going to become active. So as it is active, it requires an increase in respiration, right? It's going to need more energy as it needs more energy for movement. So more energy transferred as heat. So when you respire, you can put your hands onto your armpit right now. You can see that you produce heat, and when it's active and running about, it's going to get hotter, right? Because it's doing more respiration, more energy transferred as heat. Same as you, when you do PE, you get hot. An echinida can dilate and constrict blood vessels in its skin, just like us humans. Explain how the dilation of blood vessels, or close to the skin anyway, explain how dilation of blood vessels in the skin can help to decrease the body temperature. So how can we decrease the body temperature? So if you want to dilate, you can do vasodilation. And what that is widening the lumen of the blood vessels. Now, it's very important you say blood vessels here, because we're not talking about capillaries. They don't have elastic and muscle. They cannot widen. Arteries and veins don't do it. There are small blood vessels you don't learn about at GCSE called arterioles and venules, and they can widen and narrow. So it's better to just say blood vessels rather than naming one. So vasodilation, widening the lumen, which is the gap inside the blood vessel, allows increased blood flow to the skin. So I don't know what asked me why that went into caps, but I'm going to leave that. So increased blood flow to the skin. This allows more heat to be transferred to the environment. And for you physicists out there, by radiation, so all hot objects radiate heat out to the environment by radiation, therefore cooling the body or lowering the body temperature. You basically lose heat by radiation through vasodilation, and that would get us the marks. Increased blood flow to the skin because of vasodilation means more heat radiated, and that would be the 3 marks. So vasodilation, or widening of the lumen in the blood vessels, 1 mark. Increases the blood flow to the skin, 2 marks, which increases radiation of heat to the environment, 3 marks. Oh my gosh, of course, sweating is another way to cool down, but I'm not sure whether the echinidea sweats, but sweating would allow more evaporation, so not radiation, but more evaporation. But we weren't talking about sweating, right? An athlete trains in a hot climate. The athlete lost a large volume of water each day in sweat. The athlete's energy intake each day from food was 20,000 kilojoules. Evaporation of 1 centimeter cubed of sweat requires 2.5 kilojoules of energy. 40% of the athlete's daily energy intake was used to evaporate sweat. Calculate the volume of sweat the athlete lost each day. Give your answer in decimeters cubed. 1 decimeter is 1,000 centimeter cubed. 3 marks. Why don't you grab your calculator, pen, paper, pause a video. Okay, well, let's have a little looky here, and let's get the color up. Right, we got 20,000 joules taken in by food. 40% of the food, the energy intake was evaporated as sweat. We want to find out the total amount of sweat loss. So we want to do 40% times that. So I'm going to do 40 divided by 100, in case you don't know what that is, it's 0.4. Let's times that by 20,000, and this is going to tell us how many kilojoules were lost in sweat, and that equals 8,000 kilojoules lost in sweat. We need to turn that into a volume. So there's 8,000 kilojoules of sweat out there, 20,000 food intake. So 1 centimeter is 2.5. So I want to divide that by 2.5 to find out how many centimeters they sweated, and that is going to equal 3,200 centimeter cubed of sweat. Okay. But it tells us it wants the answer in dm. 1 dm is equal to that. So I'm going to divide that by 1,000, and that is going to get me 3.2 dm cubed of sweat lost by the athlete. Boom. Right, let's move on. No, don't tick the paper, you bozo. Right. Oh, there's another question. Suggest why the athlete was advised to take salt tablets each day. So to replace salt lost in the sweat, to replace salt lost in the sweat. Those mineral ions are very important for the nervous system and all sorts of other things. Okay, let's go. Let's move on. Right. Oh my God, it looks like tropism. Not wonderful. Students investigate the response of plants used to one-sided light. Figure 13 shows how the students set up 3 experiments. Figure 13, we got experiment A, light coming in from this side, and we got ink markings along the side of the shoot, and we have the seed. 12 hours later, the ink markings are further apart. The damp cotton wool is there. That's whatever, and it seems to have bent. Right. Experiment B is in a light-proof box. 12 hours later, it has grown. The ink markings are further apart, but it's grown straight up. It hasn't gone either way. Experiment C, it's got light from all sides, and yeah, it's done the same thing as the one in the box. It's just sort of grown. Okay, let's go down and see what the questiones are here. 9.1. Suggest 2 control variables the students should have used in their investigation. So what things would you keep the same between experiment A, B, and C to make sure they're fair? Obviously, you can't say, oh, yeah, remove the box, because they're putting the box there on purpose. So I would say it's with this cotton wool down here, the damp cotton wool. What do we need to keep the same about the cotton wool? What should we keep the same about the seeds? And etc., etc. So, and what should we keep the same about the light? Let's go smash them in there. So I reckon 2 control variables would be same light intensity. So same light intensity I would go for. I'm going to give you 3. I would go for same water volume in cotton wool, same amount of water or same volume of water in the cotton wool, the same level of dampness. I'm going to go same species of seed. I don't know, same mass of seed. What about same temperature? They didn't change the temperature, did they? Let's go same temperature. All of these might be on the mark scheme. Yeah, definitely the amount of water in the cotton wool and the light intensity will be on the mark scheme. And I reckon temperature as well, as that's a factor in photosynthesis and growth, would also be there. I don't know about carbon dioxide, but maybe the amount of carbon dioxide available and the same species of seed. Right, let's go, let's go, let's go. Maybe the same starting mass of the seeds, who knows? 9.2, describe how experiment B and C acted as controls for the investigation. So a control is where you can make a comparison. So for the investigation, we wanted to prove here that without light, the plant would, it's the light that's causing the bending. And this one is showing that when you have light from all sides, it doesn't bend. So number 1 proves that light causes the bending. And then C proves that it's the light, the direction of the light that causes the bending. When light is from all sides, it doesn't bend. So this proves that it's the light that is causing it to bend, because it doesn't bend when you have no light. But then if the light is equal from all sides, it doesn't bend. So it's light from one side only that causes it to bend. And that's what we want to put. So where are we? That's quite difficult, I think, to explain that and work that out. So experiment B shows light is needed to make it bend. It shows that light is needed from one side only to make it bend. So essentially, this proves that light is involved in bending. This proves that direction of light is important. So basically, that is what we want to do there. Right, next one. Give 2 conclusions that the students could make from the ink marks on the shoot in experiment A. 2 marks. Conclusions means what you can see. And like, just say what you can see. Don't try and explain it. Doesn't say explain, just give 2 conclusions. So let's zoom in on experiment A. We can see that the markings are close together, then they're far apart. So we can conclude from this, the fact that the markings in the top half are far apart, we can conclude that it is growing more. And we can also conclude that the growth stuff is in the top half. So let's go down. What did the question actually ask? I always have to give 2 conclusions the students can make from the ink markings in the shoot in experiment A. So from the ink markings, I can tell that whatever causes it to grow is in the top half of the leaf. The top half of the shoot has grown more. And the cells are getting bigger because they're getting stretched and further apart. So the plant cells are getting bigger. And I guess the fact that it's bending, but they might not ask that, and that it's in the top half of the plant. And then give 2 conclusions to shoot on the shoot. Oh, no, we did that. And then name the type of response shown by the seedling. So what am I going to do here? Oh, do you know what kind of response this is? Growth is mainly at the top below tip. And the reason why that is, auxins are produced in the tip of shoot. So that's the reason why. Growth is mainly at the top below the tip. Auxins are produced in the ship. It bends towards the light. So the shaded part of shoot away from light is where the growth happens. Right, let's go bish bash, and then let's bosh. The name of this process where you grow towards light in plants is called phototropism, hydrotropism growing towards water, or gravity or geotropism growing down with the force of gravity towards the ground, the center of the earth. Okay, we're on to 9.5. Oh no, not another one. Auxin is a plant hormone. Auxin is made in the shoot tip. The scientists investigate the role of auxin in the response of shoot tips to light. This is the method used. Grow 4 seedlings in the dark for a few days. Cut the tip of the shoot off each seedling. Place each seedling on a small block of agar jelly. Place the shoot tips and agar in different conditions as shown in figure 14. After 24 hours, measure the mass of the auxin in the agar blocks. So shoot tip, agar block kept in the dark, 25.3 is the mass. We've got the shoot tip kept in the dark. So that's a total of 24.9, slightly less overall. And it's got glass in between. F has light coming in from one side. So it's not in the dark. It's one-sided light coming in from there. We can see the mass of the shoot tip is greater on the left-hand side away from the light. The shaded side is greater, 17.2 and 7.8. Can add those together. That's 25 in total. And then the glass is in the middle here all the way up. And it's absolutely even, the mass on each side. And this has still got one-sided light, but the glass is in the way, G. Right, the numbers under each block show the mass of auxin that diffused into the blocks from the shoot tips. Okay, the mass of auxin that diffused in. The mass of auxin is given in AU, not the mass of the shoot. A scientist made a hypothesis. Light causes auxin to move from the side of the shoot nearest to the light, the side furthest from the light. So the auxin moves away from the shoot to the shaded side. Describe evidence which supports this. So we need to find evidence that supports this. And we can see here at F, look here, when the light is there, we can see in the tip, the auxins, imagine they're all here, they can move over here and then diffuse down over there. That's fine. So yeah, that's true. F shows that there is a greater amount of auxin on the side away from the light. Lovely. I'm going to go put that in my answer. So my evidence is light causes auxin to move from the side of the shoot nearest to the light to the. Okay, so looking at F, the auxins have moved away from the light to the shaded side. There is more auxin on shaded side. Now, I might want to give the numbers, but I can't be bothered to go back up and look, but you should give the numbers. I think it was like 17.2 and 6.8 or something like that. What was it? Let's have a little butchers then. Come on then. No, not the dreaded ticks. What were the numbers? It was 17.2 and 7.8. You can give the evidence showing that. Right, let's go. Give another scientist made a different hypothesis. Light causes the breakdown of auxin. Give the evidence from 14 that shows that this is not true. The light does not break down auxin. So what I would say as well, evidence here, you can see with G that when the glass is put there and the auxin cannot escape, it can't move away. So we know that auxin's made in the tip, and we know that if you put glass running all the way through, it cannot move over and then diffuse down like it did there. So that proves that auxin's made in the tip, G. But we can also see that when light was shining on it from one side, you still had the same amount of auxin on the light side and the dark side. So I'd put that for G. I'd put that for this answer, I'd say. Give evidence. So in G, there is the same amount of auxin on light and shaded side. I'm the side away from the light I'm calling shaded. Okay, that's just me using that as a way to describe it. The dark side, the side away from the light, okay, or further from the light, has the same amount of auxin. So that's the end of that. All right, guys, don't forget, share is caring, hit the like, punch up the bell, subscribe. Let's get to the marks. Check out my other videos.
You're still here? It's over.